All right, we will get started. Sure. Good morning and welcome everybody to the Tyler Memorial Grand Rounds at MGH in honor of Dr. Richard Tyler. Thanks everyone for being in person here in the Ether Dome and virtually. Dr. Tyler was the inaugural Chief of Neurology at Brigham and Women's Hospital and professor at Harvard Medical School. He was an exceptional neurologist and teacher, and I'm sure we have several of his mentees in the audience today. We're honored to be joined this morning virtually by some of Dr. Tyler's family as well. You guys can hear okay?
No.
No, we'll have to project. Now it's my privilege to introduce our honored speaker, Dr. Merit Cudkowicz. Merit really needs no introduction here in the Ether Dome, so I'll keep it very brief. Merit's the Chief of Neurology at MGH and the Director of the ALS program. She's one of the founders and former co-directors of the Northeast ALS Consortium, an international group of over 135 clinical sites dedicated to performing collaborative, academic-led clinical trials and research studies in ALS. She's leading the first platform trial initiative in ALS, and she's principal investigator of NeuroNext, a phase 2 network to develop innovative and new treatments for people with neurologic disorders. Her work has transformed the field of ALS, as I'm sure you'll see from her presentation today. And many of my co-residents would agree that she is a tremendous inspiration, in addition to being an incredibly humble and approachable leader of our department. So I'll turn it over now. Thank you.
Yeah, so I'm going to speak very loudly. So, but if people hear, if you can't hear me, just got a signal. And I know you can't see me. I'm on my tippy toes. So good to be back in the Ether Dome. I do confess, when I came out of the elevator, I had to look at the sign to remember which way to go. But the coffee's still here, and the desserts are still here. I don't know if neurosurgery is still paying for them, but it's really nice to be back. And I encourage people, if you're comfortable, to start coming back so we can see each other. And we'll keep working on the logistics. So it's a real honor to give the Tyler Memorial Lecture. I was a medical student and resident when Dr. Tyler was really leading the education and clinical research and clinical care at the Brigham. And he's one of the two people over there, or two of many, who inspired me to go into neurology, Dr. Tyler as well as Dr. Samuel. So this is a huge honor. And now I get to work very closely with his son, Ken, who's the editor for Animals, and I honored to be the section editor for neuromuscular. So I get to see him every Monday, which is fantastic. So Dr. Tyler actually did some of the early clinical trials in ALS back in the 80s, and they were well done studies, double-blind, placebo-controlled, and I think he'd be very happy to know the progress, though he'd probably tell us to move a little faster, that we have in ALS therapeutics. So my disclosures are here. Some consulting, and I'm a board member for Praxis. I'm not going to talk about any of those drugs or companies. So before I start, I wanted to just have a huge shout out to our ALS team and our Neurology Clinical Research Institute team. Nothing that I do is alone. It's really this amazing team of physicians and statisticians, project managers, data managers, who's really focused on finding treatments for people with ALS and helping other people in our department or other departments who want to develop treatments for their disease. I always say that it's a good sign if you could walk away the next day and all your work keeps going and the progress keeps coming. And I felt that way when Dr. Brown left for UMass. He left our ALS program very strong. And not that I'm planning to go anywhere, but I could go away tomorrow, and this amazing team would just continue this great work that we're doing together. So it's Dr. Babu, Dr. Paganoni, Dr. Berry, and Dr. Ho, and then our entire team. We definitely need an updated picture because we're about 170 people right now. And if it wasn't for hybrid work, we wouldn't fit in our current space. So that's a little silver lining. So today I'm going to talk about where we are in ALS treatments, where we're going, and how we're going to get there to really find treatments that stop this illness and eventually reverse it, hopefully even prevent it. I never thought we'd be talking about those kind of things in ALS, but we actually have an ongoing prevention trial, and we have several drugs focused on repair that are coming to trial in 2023. So huge progress in this field. I want to say that when I was a resident, I was actually advised not to go into ALS, and this was in the mid-90s as a career-ending move. It's good not, you shouldn't always listen to people give you advice. I didn't listen to that one. But fast forward now, it's a hot field, actually. It's too hot because everybody's trying to hire our faculty. But it's hot because the science is exciting and there's a real need to move fast. So I know this audience knows ALS very well. I'll just give some highlights on the kind of the numbers. There's about 30,000 people in the US living with it, more than 400,000 globally. These numbers are thought to go up probably in the next decade by 70% or so because of the population aging. So this illness, while the median age is 55, it really peaks in incidence in your late 70s and 80s. So the incidence might be 2 per 100,000 in your 50s, but it's 11 to 12 per 100,000 in your late 70s, and it keeps going up with age. The overall lifetime risk is actually not that low. It's 1 in 350 for men, 1 in 440 for women. And it is still, unfortunately, universally fatal. We know a lot more about it, about like other neurodegenerative diseases. There's a population that has a known genetic family history, usually autosomal dominant. About 10 to 20% of our patients have that. We also know that there's a big genetic component to what we call sporadic disease, people who don't have obvious family history, and that probably that illness is also counted, 50 to 60% genetic. But those are more risk factor genes that might need other things to trigger the illness. So a lot of the research has been really focused on the genetics of the illness that's given us pathways, targets, models of the disease. So a lot has been learned from that. And as we know, the very first gene in ALS was discovered here in 1993 by Dr. Robert Brown. And now there's over 50 genes that cause the genetic form of the illness. And it tells us a lot about the different pathways that can go wrong that can cause motor neuron disease. As I mentioned, this is a fast illness. I think maybe neuro-oncology is faster, but it's one of the fastest progressing diseases that we see. And it's still universally fatal. We have a long time from symptom onset to diagnosis. I know this is common in other neurodegenerative diseases, but it's a big barrier for our ability to intervene early to help patients, not just for drugs to slow down the illness, but also symptomatically. On average, it's a year, and that is mainly because people are going to non-neurologists. They often start with orthopedics or neurosurgeons or ENT. And on average, before we see patients here, our patients have seen 3 or 4 other non-neurologists. So there's a big effort to do kind of more education campaigns, as well as to develop kind of fancy, blood or CSF diagnostic markers. But a lot of it is actually just knowledge and experience of other physicians. So the prognosis is usually 2 to 3 years, but there's a huge range. And one thing we don't do is tell people how long they're going to live when they see us. We often have people come to us for second or third opinions, and they've been told, to pack up your affairs, you're going to live 2 or 3 years. And the bottom line is we don't actually know. We don't have good prognostic markers yet. And some people live 10 to 20 years, some live 6 months. And until you followed someone for a while, we don't really know their disease course. We do have things we can do. That's the other thing we hear often from patients, that they were told that there's nothing to do. There's actually a lot to do. So we know that people who go to multidisciplinary care clinics, like the one we have here, live longer, have better quality of life. We also have 3 FDA-approved drugs. 2 of them slow the decline, and 1 of them is a symptomatic treatment. And there's, last count, over 140 clinical trials going on in the world. The good news for us is only 14 are happening here, but it is a busy clinical trial time, and we're trying to, again, model ourselves after some of the oncology field where everybody who comes in is offered or has an option for a clinical trial. The genetics, as I mentioned, have given us lots of clues into the underlying biology, and those 50 different genes go into different categories. There's many that are on RNA metabolism. There's others on axonal degeneration. We know if we look at the tissue of people with ALS, whether it's blood or autopsy, that there's a lot of inflammation there. So this is just a list of the abnormalities. There's a very complex biology. What we don't know is what's the most upstream thing that happens. And also, is the biology different in different people? At some point, there's a common pathway. For example, we know everybody that we see has markers of inflammation, but we want to get more targeted so that we can get maybe more effective drugs. It's classically, as you know, called a motor neuron disease with upper and lower motor neuron disease, but the genetics have also told us that it's much more than that. We have genes that cause both FTD and motor neuron disease. And now again, that more people are studying this and seeing the illness, we know that they have autonomic dysfunction, they have sensory loss, but it's still predominantly their symptoms are mediated by the upper and lower motor neuron disease dysfunction. So where we are, we have these two marketed drugs, riluzole, which has been here since the 90s. This is thought to work by blocking presynaptic glutamate release. It was developed without any animal models. So sometimes when people say you need all these animal models or your drug to work in there, a reminder that we actually don't know how predictive our animal models are. This drug does, prolong life about 10%. It's fairly safe. Every trial that's tested another drug, when they go back and look at people on or off riluzole, show the same 10% effect on survival. So it is really standard of care, and about 80% of our patients take this. Now, in 2017, an antioxidant called edaravone, also known as Radicava, came on the market. And this drug initially came out as an intravenous formulation and did not have a big uptake because it was 10 days a month, required a port, and inconvenient for patients. It was just approved literally 3 weeks ago by the FDA in an oral formulation. All they had to do was show equivalence. And now our phones are ringing and epic charts are buzzing of people wanting to be on this drug. So this came again with not a whole depth of science. There was like one mouse study that showed some protection of motor neurons, but there were two studies done in Japan. The first one was actually negative. It took a broad range of patients and followed them. And when they did some post hoc analyses, they found that if you started early in the illness, they could see a slowing of the loss of function. Now, post hoc analyses are usually wrong, and when you repeat them, they don't show up positive. But this time, it did show up positive. So they repeated a study again, all in Japan, of in people 18 months in the symptoms with rapid progression. And they picked that population based on the post hoc analysis, but also to be able to get an answer in 6 months. And they showed a 33% slowing of the loss of function then. And based on that study in Japan, really one pivotal study, it got approved in the United States. And that was a game changer for the FDA regulation here. And it was in response to a lot of patient pressure to allow people with ALS to have a little more uncertainty or take a little more risks, but have access to drugs faster. So that was approved, but provisionally that the company had to do another phase 3 trial of testing actually everyday dosing versus 10 by 2024. And that study is ongoing. And in addition, a competitor company has a tablet form that is being tested in Europe against placebo. So in a year and a half, we'll have I think more certainty about whether this works. But in the meantime, it is something that we can prescribe our patients. And we are, again, if patients want it, starting to give the oral version of it. It doesn't have any real significant side effects. A few people can have some loss of balance in the first cycle, and that requires stopping it, but nothing permanent. The other marketed drug is Nuedexta. I think we all know this as a treatment for pseudobulbar affect. That's what it's on the market for in ALS. In those studies for pseudobulbar affect, our patients were telling us that their speech and their swallowing was better. So with our network, we did a study to look at that, and we did find a positive study that about 75% of people with ALS will have quantitative and clinically obvious improvement in speech and swallowing, but it's not marketed for that. It's marketed for pseudobulbar affect. So that's what we have in our armamentarium, plus, of course, multidisciplinary care. But we want to do much more. And we really do think we're going to need a cocktail approach, at least until we understand what maybe the most important or most upstream biology is. We're also, as I mentioned, starting to think about prevention, and this is in the genetic forms of the illness. So we, again, led here by Dr. Berry and Dr. Nicholson, have started, big cohorts to follow people at risk for the disease, to understand what's the earliest biologic or clinical change, but also to prepare for clinical trials. And there is now an active gene therapy prevention trial in SOD1 ALS that's ongoing. And lastly, as I mentioned, we're starting to think about how you can return some function, which sounded crazy, but there are, there are people working on this in spinal cord injury and stroke. And so we're trying to attract those scientists to the ALS field. And then at least in 2023, the first drug in this pathway, which is a SARM1 inhibitor, will be tested in people with ALS. So that's where we are. This is where we're going. And this list is not complete, but there are now at least over 250 companies working in the ALS space, and then thousands of investigators. And we're starting to see some positive results. In green are 2 clinical trials that in late stage testing with positive results. They're both under regulatory review in some country. I'll tell you a little bit about the AMYLX one, the first one. The ones in blue, tofersen is a gene therapy for SOD1, Neuron is a stem cell trial. They both have biomarker positive effects, but less clear clinical effects, a little bit like the aducanumab story, but I think with a better probably ending, more hope. And then the ones in red were negative. And then everything else is kind of ongoing. And I said, as I mentioned, there's over 140 trials, so this is just a snapshot of them. But because the pipeline was so big, we really started to think that if we do one at a time, we'll never get through the list. And we needed more innovative ways to screen more rapidly through these possible drugs, especially really in the absence of having full knowledge of the biology. And that really led us to create the first AI platform trial in ALS, which I'll tell you a little bit about. And again, that's a model of being efficient that we've copied pretty much from oncology, particularly breast cancer and lung cancer. So I'm not going to tell you too much about methylcobalamin, so I'll just highlight it here that this is a form of B12, again, an antioxidant that also was developed in Japan. And it's almost the same story as oral edaravone in that the first study took a broad group of patients and was negative, but a post hoc analysis showed a 50% survival benefit if you started it in the first 15 months of your symptom onset. And based on that, Eisai, that owns that drug, did another study, which they just reported out in JAMA Neurology about 2 or 3 weeks ago, where they only gave it to people in the first 12 months of symptom onset, and they showed a 43% slowing of loss of function. So that, I think, is going forward for regulatory approval in Asia. I don't know the plans yet in the US, but that is, again, a hopeful a new advance and something that we might be able to give our patients. So I wanted to take a moment to just talk about disease networks, because when I was a fellow, Ann Young kind of shoved me literally, I think, into the Huntington study group and said, go learn from them about how to lead study groups. And she put me on all their steering committees, and they were very tolerant of just having me there and listen, and watched how they really got the whole community together to work on science, biomarkers, clinical trials together. And we then started a similar initiative in ALS, and it was 9 centers. We actually used to meet at the Brigham because Jeremy Scheffner was there and he was the leader with Bob Brown, and 9 other centers. And we knew nothing, like we had no outcome measures, no biomarkers. We just knew that a couple of genes had been discovered and maybe we could find treatments. And this has been a great network for really collaborating on a relatively rare disease, and at a time where there weren't a lot of resources. So we're now over 140 centers. I stepped down as chairing it when I became chair of the department here, and it's successfully gone through a couple transitions of chairs, which is also a really nice thing that it's a very solidified network. We've done over 57 studies together. It's scientists, it's healthcare providers of any field, anyone who touches ALS is part of this. Every trial we do, we insist that the placebo data is then donated to an online database called PROACT. So they have over 20,000 people's data in there, and lots of papers have come from that. Every trial also, so the biofluids have to be shared, and those are housed here and then Barrows and led by Dr. Berry, and have led to some of the discoveries of some of the new biomarkers we have around neurofilament, as well as measures in urine. We also created a central IRB for the network so that we could speed things up. And we've trained really thousands of people in ALS with the goal, again, is to have many people that can design and lead clinical trials and many sites that can enroll participants close to their home. And the last thing is that we have a science advisory board that has scientists from all over the United States who meet with companies and give them advice both on preclinical studies as well as clinical trial design. And I think every month we're meeting with 3 or 4 companies. And so that's a very nice way, again, to kind of work as a community to try to develop treatments for this illness. So because of that network, we were able to really help this company, Amylyx, develop their treatment. And this is one of the positive drugs that's under FDA and Health Canada and EMA review right now. And the trial was led by Dr. Paganoni. It's her first trial. It's a good way to start. Though the only problem is, might be the only way is to go down or stable, but it's a great start. And I just want to have a shout out to Rudy, because if it wasn't for Rudy Tansey, this drug probably wouldn't exist. The idea for this drug came from 2 undergraduates at Brown University, Josh and Justin, who were thinking about repurposing old drugs for Alzheimer's disease. And they thought about TUDCA as a drug supplement that could work on mitochondrial dysfunction, and sodium phenylbutyrate, an HDAC inhibitor that could work on ER stress. And they brought their idea to Rudy, and Rudy does say that he only answered them because they were in the same fraternity he was when he was in college. And I believe him, and I don't know, you guys don't know me that well, but I used to always tell my daughter, you cannot join a sorority, you gotta study, blah, blah, blah. So I did call her up and tell her that it was okay. Good networking. Anyway, so he helped them with their preclinical work, and I met them by chance and diverted them a little bit from Alzheimer's to ALS, but they're still working with Steve Arnold in Alzheimer's disease. And they did show that in cell models of neurodegeneration and in the animal model of ALS, that the 2 drugs protected motor neurons alone, but they were more than additive together. And then they came to our NEAL Science Advisory Board and presented the idea, and that group helped them with the science as well as the design of the trial. And we launched the study in 2017 at 25 centers in our network. We also helped them write the grants and get, this was largely funded by foundations at that time. So the design really mimicked the second oral edaravone study. So whenever you have one success, there's a tendency to repeat the same design, but it took people early in the illness, 18 months from symptom onset, and fast progression. So we defined fast progression by something called definite ALS, which means that you have ALS in 3 body regions, bulbar, arms, and legs. And if you have all that in 18 months, that's pretty rapid. So 137 people, 2-to-1 randomization, treated for 6 months, and then put in an open label extension period, but they remained blinded to what they were on during the double-blind period. And that's important for being able to also make some conclusions from the open label extension phase as well. This is the primary outcome measure, and it did hit its primary outcome measure. It slowed the illness by about 25%. This is a rating scale that goes from 0 to 48, and 48 being normal function. And typically, if we weren't trying to find a fast progressing group, people drop 1 point a month on this scale. But as you see in the placebo group, they dropped 1.6. And again, that was by design to try to get an answer in 6 months. And for patients, you ask, what does this mean? It's meaningful to have preservation of a point or so a month. That means being able, for example, to walk up the stairs without holding the railing, or be able to feed yourself. So they're significant points. Now, it wasn't powered for the secondary outcome measures. And what I mean by that is when we look at strength and breathing, it's more variable within a person, between people than these ALS functional rating scale. So we would have needed to have more in the 200 range to see success on those outcome measures. But we did see everything going in the right direction for strength as well as breathing function. And fortunately, again, in a 6-month study, we didn't see people, you didn't see a lot of deaths. As you see in the first 6 months here, the 2 curves are on top of each other. But when we looked at people who started at 6 months earlier to people who started the open label 6 months later, the people who were on placebo initially, we did see a survival benefit of about 4.8 months. And so that was going back to the importance, at least in these rare diseases, of the open label data and being rigorous with it. And keeping people blinded to what they were on the double blind during that period. And in a way, this could be considered a confirmatory, study to the first one. Now, when we designed it, we had no plans that this would be the only study ever to happen. We designed it as a phase 2 study, but because of the enormous patient pressure on the FDA, and because there are actually 2 old drugs that don't have significant safety issues, was really very well tolerated. There was a groundswell of kind of patient and foundation advocacy pressure to the FDA to consider approving this. When we met with the FDA, they did tell us to do a phase 3 trial. And so that was designed, and that's called Phoenix, and that's an ongoing phase 3 trial that's global, 600 participants. It's a little longer, so 48 weeks, and it's a little broader inclusion criteria. And that is about halfway enrolled and should read out again another year and a half. But in the meantime, the FDA did call the company and ask them to submit a new drug application, and that was last July. And that happened. And they had an AdCom meeting in March, and the AdCom actually voted 4-6 against approval. And again, this was the first AdCom after the AdCom for aducanumab, so it was a highly watched AdCom, and we don't really know what the FDA is going to do. They were supposed to make a decision by end of June, but they just asked for a 3-month extension so they could review additional survival data. So we'll have an answer about whether this is going to be approved in the U.S. By end of September. We should hear sooner than that from Canada, and by the end of the year from Europe. But no matter what the decision is, there's a commitment to finishing this trial so that we will have the results. And so this comes down to like a philosophical debate in the ALS community about whether for a fatal disease, you can take a little uncertainty and have this kind of provisional approval, have access for people living today while you do a confirmatory study. And I, thanks to Ken Tyler letting me do it, I wrote an editorial about the AdCom because I personally thought they got it wrong. And I don't have any money from Amylyx, but obviously I'm invested in this. But as someone who sees patients all the time who, who die, basically, I do think that the risk is greater of making them wait 2 years, which case everyone we're following today will be dead, and then finding out that the drug actually is positive. That's a bigger risk than letting people take a safe drug now, and then maybe in 2 years we find out that it didn't work. But again, it's a debate in the ALS community right now. So maybe a little less controversial or different controversial is the first gene therapy result, and this is so exciting for so many reasons. But I'll say that Susan here was the very first patient I ever took care of in 1994 as a fellow with Dr. Brown. The gene had been just discovered a year before. Her family had the SOD1 gene, and 3 of the siblings, all in their 40s, came down with SOD1 ALS at the same time. And with Dr. Brown, I took care of them, and they and enrolled them in my first clinical trial, which I'm slightly embarrassed to say was the opposite experiment of this one, where we actually gave people SOD1 because we didn't know back then how the mutations were working, and that it was actually a toxic gain of function and not a loss of function. But I did learn a lot about how doing clinical trials and a lot about how to take care of people with ALS. We really thought back then that we were going to be able to cure SOD1, quickly, but it took 2.5 or so decades to get to the point where gene therapy was possible. And so it was very exciting to really be part of the first antisense oligonucleotide treatment for a familial form of ALS. So to person's name the drug, it decreases SOD1 protein levels. It's not mutation specific, so it will decrease both the wild type and mutant SOD1. Lots of preclinical work showing that this can save motor neurons and prolong life in animal models and in cell models of SOD1 ALS. We did the phase 1 trial a couple of years ago, and we published it in the New England Journal of Medicine. The lead author is Tim Miller, a colleague of mine from WashU. And this was his first trial too. He's a lab-based scientist. But a lot of the work for SOD1 came from his lab. We showed target engagement. It lowered SOD1 levels in the spinal fluid by about 36%. We know from the animal studies that that's equivalent to about 90% in the spinal cord. So that's what we were trying to achieve. And clinically, even though with small numbers in this phase 1, the brown line are the people with the fastest form of SOD1, A4V, really not progressing in 12 weeks, whereas the placebo group was doing as expected, which was going down very fast on this range scale. So we were very excited about that. We saw that also in breathing. We had modified the phase 1 to a phase 3. So this was the fastest transition I've seen from phase 1 to phase 3. It was the same protocol, and then expanded it to many more patients. Now, sometimes our patients ask us, like, why are you spending all this time on the 10% of people with ALS and not the rest of us? And one of the answers is that that's not how it works. Everything we're learning from the genetic form helps us for the sporadic. And the gene therapy is a good example because the approach of being able to modulate proteins up and down is now going forward in sporadic disease. So there's an active trial of an ASO against ataxin-2, lowering ataxin-2, which has been shown in the lab to promote axonal growth. And also based on Clotilde's work here, an ASO for increasing staphynin-2 should start this year. And none of that would be possible if we didn't learn about these gene therapies from the familial form. There's also active gene therapies in 2 other genetic forms, FUS, which is a very rapid, usually young onset genetic form of ALS, and C9orf72. There's 3 different gene therapy trials going on there, and that's the most common genetic form of ALS and also causes FTD. So a lot of activity in the gene therapy space for ALS. And our team, and mainly Dr. Babu, has really been the leader in seeing the patients with genetic forms and treating them with these different gene therapy approaches. So the Jefferson study is a little complicated, but I think it's very exciting. Last week at the ANCALS meeting, Dr. Miller presented the updated results for both the double-blind and the open-label extension of the phase 3 part. Now, we had previously announced that the phase 3 double-blind part did not hit its primary outcome measure, and that's still true. But this is an update of what happens to people after they continued in the open label, so that kind of delayed start versus starting from the beginning. And I think you'll find this exciting. So just to repeat, so for the double blind, it was 28 weeks. We picked the highest dose from the phase 1. We took we enriched for fast progressing patients, but also took some slow progressing patients. And then everyone could roll over into the open label extension. Now, because it's a super rare form of a rare disease, it took a long time to enroll 100 patients. So some of the people in open label have been on it for many years. And also the people in the phase 1 continued. And again, clinically, and I don't believe in anecdotes, but I have to say that we saw people doing amazingly well. There was someone I take care of, whose mother I took care of. Her mother died in 9 months. She got sick, taking care of her mom, was one of the first patients in the phase 1, and we're still taking care of her today. And it's not that it stopped, but she's lived 5 years right now, which we just don't see in that illness. So there's a lot to learn about timing and dosing, but I think there's a lot of hope here. So I'm going to show you data from the 12 months where everybody in the open label has finished 12 months. So again, 108 people, it was 2-to-1 randomization, and almost everyone finished the 28 weeks. There were a few deaths, and then about 88% of them went into open label extension. I know this is a busy slide, but I'll just say that we did have, for a rare disease, a fair amount of data. So just to show again target engagement. So green are people who started on active drug in the double blind. The dashed line blue is placebo. And then in the 28 weeks, they go to open label. And you'll see that again, we lowered the SOD1 level about 33%. And then the people in placebo, it stayed stable. And then it went down when they went in open label. So again, hitting target engagement. We had a huge effect on neurofilament, and this is really the only drug in ALS that we've seen have this type of effect on this marker of axonal loss. Now, we don't know if this is a good surrogate marker in ALS. We're hoping it is, but it hasn't been really proven. But anyway, there was a dramatic lowering, about 50% in the spinal fluid on people who started Jefferson early in green here. Again, stable, which is what we tend to see in people, that it starts elevated and then stable during their course. And then again, a dramatic drop when they went into open label extension. So, when you see this, you're thinking huge success. But again, on the clinical scale, the ALSFRS, in the double blind period, this was not statistically significant. It trended favorably, but not what we were hoping for. But then again, the green are people who started on the drug and then continued on the drug. The blue are placebo to drug. And you start to see over time more of this kind of flattening and separation, which with the people who started later slowing down, but never really catching up to the people who started earlier. And I'll just show you all the measurements. This is the breathing function where we see that same type of slowing over time. And I think this really tells us for gene therapy trials, maybe 6 months is not long enough, that you need some time, both to lower the target of interest and to have an effect. And then this is the effect on strength measures that we did with a handheld dynamometer. And, without over exaggerating what we're seeing, you certainly looks like maybe people are even getting some strength back over time. So we're thrilled about this. We don't know what it means as far as regulatory, because again, the primary double-blind period did not meet its outcome measure, but we certainly have an effect on neurofilament and then maybe a longer-term effect clinically. Again, there weren't a lot of deaths in the first 26 weeks. The groups are pretty much similar. This is again showing that over time, it's not a cure. People are still unfortunately passing away from this, but there is a separation between those who started early versus those who started later. Now, this wasn't a totally benign treatment. Big picture for fatal disease, I think it's okay, but we did have two patients who had myelitis. They both did recover, but that was a serious side effect. So just to summarize on this, it does take a couple weeks to lower the target, a little longer to lower the plasma neurofilament. We started to see some trends clinically at 28 weeks, but it really took much longer and in the open label part to see that bigger effect on clinical function. So there's still a discussion about the next steps, whether we need another trial or if this is enough for FDA submission. Again, this is 2% of people with ALS who have this particular mutation. So it's not unlike the aducanumab story in Alzheimer's. This won't necessarily bankrupt Medicare if it does get approved. Now, at the same time, we're learning that maybe we need to go in earlier. Now, we looked at that in the Traversion study, if that was one of the reasons it didn't work in the 6 months period, but we couldn't really see anything significant about going earlier versus later. And it might just have been too small a group. But there is a prevention study that has started. It's led by Dr. Benatar at Miami. We're one of the sites in that. And here people are followed who know that they have the gene. They're asymptomatic. They're followed routinely with the blood tests for neurofilament. And he's shown in his pre-symptomatic trials that about a year before you get clinical symptoms, your neurofilament level spikes up. So in the trial, when you reach a certain amount or you increase by a certain amount, you're randomized to drug or placebo and then followed. And this is going to be a study that's going to take about 5 years, but it has started and we are actively looking for gene carriers. And I hope this will be a model for other prevention trials, not just in ALS, but also in other neurogenetic diseases. So my last couple minutes, I want to talk a little bit about the platform trial. And I know that for those of you who were at Sabrina's talk in February, where she got the Lisa Gravickas Memorial Lecture, she talked a little bit about this. So I'll give a little bit of an update. So we really decided around 2018 that we thought that we were ready for a platform trial approach in ALS. We met initially with all the groups that are doing that in oncology, and they were very generous in their time, like the statisticians, the lead investigators, and wanted to learn what worked for them, what didn't work for them, but also for them to kind of look at our field and say, are we ready or not? And it was really a resounding yes, that we had the networks, we had the outcome measures, we had the pipeline and the statistical knowledge to do this. So, what is a platform trial? So traditionally we test one drug at a time. It takes a year to get all your sites up and ready. You do your trial. If it works, great. You go to the next trial. If it doesn't work, you take the whole network down, everything that you've built, and you start all over again. It's terribly inefficient. And we wouldn't do this in most other things that we do in life. And the analogy is often with a sports stadium is you don't build it, play a game, take it down. You really build it and you keep going until you get the championship. So we wanted to do something like that in ALS and build the infrastructure to keep running trials until we found really effective ones. And you can go, as high as you want on the number of trials. I think the breast cancer group, the iSpy group, did maybe like 14 or 15 drugs at a time. But everyone kept the one advice they told us is start simple. So we started with 3. And we couldn't have done this, without Sean Healy. And I wanted just to take a moment about what an amazing person he was. And our center is named after him, but he loved this idea. And he gave us the money to start it. And because of that gift, we could get this off the ground really quickly. Because a lot of other groups who want to do platform trials spend a lot of time designing it and then trying to raise the money. And in a way, we had the money and then we could design it. And that was really fabulous. There's been a lot of studies about platform trials, and basically they cut time to getting to effective treatments in half. They cut the costs by about a third, and they greatly reduced the number of people who are on placebo because you share the data from placebo between different drug regimens. Sandy, who's a patient who I met online, who I've now met at many conferences, is a strong advocate for research. She's an amazing woman, but this is really her quote, that she lost the privilege of working on the human time clock when she was diagnosed, and the ALS clock is faster and is always pushing us to move fast. And she loves this idea. She's on our advisory committee, and it's really helped us get it enrolling very fast. So what we did is we said, all right, we have money to do this. The first 4 companies that work with us will pay half of it for you. After that, people have to pay their entire way. And we made it a competition to be in the platform trial. And we got 33 applications from all over the world from companies, and we picked 4. And we had our science board from Niels pick them. You know, we'll see how good we are at picking them. We're going to have the results of the first 4 this August and September. But we tried to also pick different pathways. Zilucoplan is a complement 5 inhibitor. For Dipistat, works on microglial activation. CNM-AU8 is catalyzes NAD formation in the motor neurons and has been shown to decrease TDP-43 aggregates. And then the 4th one we added, pridopidine, is a sigma-1 agonist that blocks nuclear pore dysfunction. So they're all different. They all had great science, data in multiple models, knew the dose, had proof that they could get into the brain. And so we first started with 3. And how it works for people who come into it is they're randomly assigned to one of the available drugs. So the participants don't get to pick which drug that they take. So that is different from when you just have a single study. Then there's a second randomization, and we pick 3-to-1 drug versus placebo. And then they're treated for 6 months. And then there's an open label extension. So if you're in zilucoplan, you go into the zilucoplan open label extension. And then in the end, we look at all the data from, let's say, the zilucoplan group, and we compare it to the shared placebo. So you're still getting a 1-to-1. And sometimes actually you have more placebos than active drug comparison. So you still have a lot of power to tell if your drug works or not. And we picked 160 and did a lot of modeling, and we'll be able to pick up a 30% or greater slowing in the disease progression with this size and this duration of treatment. So where you get the efficiencies are on the shared placebo. So you have fewer people that you need to put in your arms, but you also get the efficiency in like startup. So we started with 3, but 4 months later, we added regimen D. And to add it, all we had to do was amend the protocol. So the FDA asked us for 30 days, and they did it in 30 days. And the IRB approved it in 9 days. And the reason they approved it in 9 days is because they're only looking at the safety data. They've already approved the protocol and the design and statistics. And when we added the 5th regimen, that was approved by IRB in 11 days. And since we're using a central IRB, essentially all your sites are activated. So instead of what typically happens, where it takes like a year to get everybody off the ground, this is incredibly efficient. So you're saving money and time that way. So I'm a big fan of this. It takes a team, and I don't think we could have done this without the NCRI here and all the 2 decades of experience in running clinical trials in ALS and our network. So the other thing that really helped this get off the ground was at the beginning, really getting everybody's buy-in. We started with the patients, and because of that, they've given us all sorts of awards. This trial enrolled 3 times faster than any other trial. It actually, in an odd way, overwhelmed our sites. Our sites could not keep up with the demand to be enrolled. And so we're working on that. It's a good problem to have. We met with the FDA twice, and they were bought into it. They loved it. And they've been very helpful and really granted us as many type C meetings as we want. We worked through a lot of the issues that, even though there have been other platform trials, are not fully worked out. For example, if you're always using the placebo data, how much of that placebo data can you actually publish without without impacting its future use? Or how do you share that kind of data? So they've been very helpful on that. We got all the foundations involved to the point that really, like, they all wanted to fund us as well to be part of this, and that's been helpful because we needed additional funds. And then we had to do a lot of education on our site investigators because most of us had no idea anything about platform trials before we started this. So just briefly, for this to work, you have a master protocol, and you have to, each of the regimens has to do the same thing as far as like the endpoints you're doing, the how long you're treating it, how the visit schedule, You can have some flexibility on which primary outcome measure someone wants, but you have to collect the same outcome measures. Otherwise, you don't have the ability to share the placebo. So there is a loss of a little bit of control for the companies that are working with us, but that's one of the key features. But then you can keep, like I said, adding regimens, and you can have some things that are specific to a drug. For example, their dosage, the route of administration, something related to their safety profile. But we really try to keep things as common as possible between the different regimens. Let me just move to this. So as I mentioned, it's 6 months, 160, but in the end, it's 120 to 160 comparison. We have about 90% power to pick up something 30% or greater. We took a broader group, and we'll find out in August whether this was a good decision or a bad decision. But the reason we went back to a broader group is because, again, the patients were saying, great that edaravone and Amylyx worked, but that's 7% of the population that you're including in trials. And what about the rest of us? So we did do a lot of modeling with that PROACT data set of 20,000 people to see if we took a broader group, can we still pick up a signal? And that guided us on our sample size. And so we do think that we can. But again, if one of the drugs or some of the drugs work better early, we might miss it. So the nice thing again about the platform trials is you can adapt as you get data to see if you need to do it differently in your next group of drugs. We're looking at the same endpoint, ALS functional rating scale. We have some crazy statistics that I won't be able to describe to deal with the placebos being of different route of administration and perhaps different time. But that's all been approved by the FDA. We're looking at respiratory function, strength, and survival. And the other good thing about the platform trials, you can add in a lot of exploratory endpoints, and these data belong to the community. So again, sometimes when we're doing trials with companies, if the trial was negative, all the data belongs to them, the samples, the data, and maybe they'll do something with it, but most often they don't. They just move on. And so we designed this and we did all the contracts with it so that all the samples belong to the NEALS network and all the placebo data belongs to the NEALS network. So again, we could do that because one, we were paying for the half of it, and because again, we have this infrastructure of a network that could say, this is basically how we're doing it. And so I think that will help. So we've added a couple exploratory endpoints like home measures of breathing, home measures of voice, different fluid biomarkers, and hopefully no matter what the outcome of the actual drug, we'll keep learning about the biology of ALS. So we published the paper in Annals and we made it a free download, which as you guys know is not like a low cost thing, but we thought it was important because we learned so much from these other groups that were doing platform trials that we wanted to get out there what this protocol looked like, some of the key learnings that we have. And we have a similar one for statistics that we're submitting, dealing again with those complex things with platform trials. And the master protocol is also available online. And we've been trying to, again, pay it forward because we got so much help from so many groups that we've met with Michael J. Fox about their Parkinson's plans for a platform trial and also different groups in stroke and TBI. So I think we're going to see more and more of these in neurology as we get more and more therapeutic options coming our way. So I mentioned that our sites were overwhelmed, and they really were. It was, again, it's a unique situation because usually we're having a hard time filling our trials. And here our patients were calling and saying, listen, I'm trying to get into UPenn and they tell me it's a 3-month wait. So we did a couple of things. One is we expanded to 74 sites and we are trying to create some new sites in areas that are not covered. But we also developed the patient navigator program, and that's with Catherine Small and Allison Boulad. Allison's husband had ALS, and she's a strong advocate in the community, and Catherine works here at Mass General. And they are basically available for patients, and they'll help find you a center that might have a spot. So we, for example, have 3 sites in Philadelphia. So if UPenn can't see them, they'll get them in Jefferson or another one. And that's been enormously helpful. The other thing our patient advisory group said is have a weekly webinar. Just let us know how it's going and let us have a chance to give feedback. And so we do that every Thursday at 5, Sabrina and myself. And it's been fantastic. And I think these initiatives kind of help get the word out that trials are important and that we need to enroll. And I really do think that we've gone from the days where maybe 10% of people with ALS were in trials to maybe now we're maybe at 30 or 40%. We still have a ways to go, but this is an extremely helpful approach. So we finished enrollment in the first 4. Now regimen A, which was the Luca plan, we did stop early in January for futility. So the other thing we built into this was every 12 weeks, interim look for futility after the first 40 people had finished 6 months. And we did that because we didn't want to waste our time, our money, and more importantly, the patient's time. And so regimen A did pass that futility boundary. And so we took everybody off of that in January. The other 3 have passed all their futility looks, and we are expecting results from B and C in August and D in September. So a bad timing when you're doing trials to have your results come out in August. But we're planning to work on them all, and hopefully 1 or if not 2 or 3 will work. And again, then the beauty of the platform trials, you can adapt. Like if one of them works, again, assuming no safety reasons you can't do it, you start adding them to the next regimens. We started enrolling in E, which is the Trehalose, which is a drug that works on autophagy. We're designing F, and we're in discussions with about 2 other companies as well. So our goal is to kind of keep adding 2 or 3. I think we found out that 5 at one time is probably too much for our sites with their current staffing model. And that maybe is for another discussion one day. I do think this idea that physicians spend 5% of the time enrolling participants as a site investigator isn't going to work. We probably need another model of if we're going to have this many trials, maybe again, we can learn from oncology of how do you have an efficient trial network. So I'll just conclude that I hope I got the message that this is a super exciting time in ALS and in the broader neurotherapeutics, and the pipeline is big. We're starting to see some success, and I hope, again, that'll translate that we're eventually a bit like multiple sclerosis, where we have a lot of options for our patients. We have a really engaged patient community, and it makes it fun to work when you feel like you're working with each other as colleagues, but you're also working with the patient community to solve the same problem. At this point, our biggest barrier still, I think, is understanding the biology. While we understand a lot more, there's huge pieces of the puzzle that are missing, and that's why we have people like Gaz here, and Clotilde and Brian and many others working on that. As I mentioned, I'm super excited that we actually have a prevention trial going on, and then people are talking about how do you return function. So lastly, I just want to again shout out to this amazing network of Neil's investigators that we're super excited to be meeting in person again in October. We haven't seen each other in person since 2019. And the many foundations that support the platform trial, but a lot more of our work as well. And the companies that took the step to work with us and be the first ones in this platform trial. So thank you. I hope I left a couple of minutes for questions. I think I might have just shut down the Zoom. No, I didn't. Yeah, I did.
Well, we can start with any questions in the audience if there are.
Yeah, Marcella, sorry about that.
That's okay.
Absolutely fantastic. Now I know how you've tackled the department issues that are very challenging with that courage and mindset for complexity and team building. So my question is related to clinical phenotypes and how they have informed results of clinical trials, either more restricted forms of motor neuron disease, or even when you say you had other presentations outside the
Yeah, it's a really good question. So far, we haven't really seen that certain clinical phenotypes, that would say bulbar onset versus like limb onset, that they respond differently to drugs, or that they have different biomarkers. So at the moment, the field